Hi, this is the protein processing and regulation lecture. So remember, this is chapter 10 in the new version of the textbook. All right, so in terms of what this will cover, translation. So we obviously, up to the last, what you went up to in the last exam, we talked about transcription and a lot about regulation of transcription. So we'll also, we'll talk about translation, the process of translation in general. But then we will also talk about regulation, a little bit of regulation of translation, but mostly more so regulation of protein function in general, okay? And so protein degradation sort of fits in with that in terms of regulation. Obviously, if a protein is degraded quickly, then it will, not be able to function once it's degraded. Proteins that would be able to hang around for a longer amount of time would think of them as being sort of more active. We'll also talk about protein folding and processing of proteins. Okay, so we all know that translation is the synthesis of proteins, and that it's directed by an mRNA template. Remember, in transcription, inside the nucleus, right, we produce mRNA from DNA, and then that mRNA is transported out of the nucleus and essentially joins with a ribosome, and translation is going to occur. Okay, and so that's where we're going to form that polypeptide chain, and then those chains have to be appropriately folded. So remember, we talked about primary, secondary, and tertiary levels of protein structure, okay? And so folding is one thing that has to happen, but there's also other processing that can happen to the proteins as well, okay? Then they have to get sorted and transported throughout the cell. So it says gene expression is regulated at the level of translation, right? We talked about regulation of gene expression on the level of transcription, and a lot of it occurs on the level of transcription, but there is still some regulation during translation as well, okay? And so again, other controls in terms of like phosphorylating a protein, we sort of talked about that a little bit as well. Okay. All right, in terms of how those mRNAs are read during translation, right? So again, we're talking about, we mentioned transcription happens in the nucleus. From DNA, you get an mRNA transcript. Remember that mRNA transcript gets processed in the nucleus, and then once it's ready, it gets sent out. And then again, we'll find a ribosome attached to the ribosome, and translation will occur. The way the transfer RNA, or the way translation is going to happen, again, is in the 5' to 3' direction, okay? And those polypeptide chains are always synthesized from the amino to the carboxy terminus. Okay, so, and we may remember some of this basic, hopefully we remember some of these basic ideas, but obviously we're going to elaborate a little bit more on them. So each amino acid has a specific codon, right? That's composed of 3 bases. That's in the mRNA. And remember, it has to be read in that 5' to 3' direction. Ribosomes, right? Transfer RNAs. We haven't mentioned them I haven't mentioned them just yet right now. We've talked about them before. They are a necessary component of translation as well. And basically, you have interactions between the 3 types of RNA in protein synthesis or translation, right? So the mRNA is going to provide those codons. The tRNA, right, has the anticodon and the amino acid attached to it, and the ribosomal RNA is part of the ribosome, okay? And then there's other proteins involved as well. Okay, so let's talk a little bit more about those transfer RNAs. And so it says they align amino acids with the corresponding codons on the mRNA template, okay? So they're themselves, in terms of their structure, about 70 to 80 nucleotides long, and they have this sort of characteristic clover leaf or kind of a lowercase t shape. And the reason that shape forms has to do with basically just base pairing between the bases that make up the tRNAs. Okay, so if you look at that here, this is what this is showing you, right? So again, this is RNA, right? It's nucleotide sequence, just like we talked about before. But just because of the sequence and complementary base pairing, you get this sort of secondary structure that forms, okay? And so if you just look at the basic structure of that tRNA, as we said, sort of clover leaf or a lowercase t shape, and you can see here is this, at that 3' end, right, that's where the amino acid is actually going to attach to. Down here is the anticodon that's going to match up with the codon in the mRNA. okay? So this is just showing you in another way the structure as well, and same thing here. Okay, so it says they fold into these compact L shapes to fit onto the ribosomes during translation. They all have a sequence, a CCA at the 3' end, and that's where that amino acid is going to attach, okay? That anticodon loop, like I showed you in that other figure, will bind to the appropriate codon via complementary base pairing, right? I showed you that already. So in terms of specifically how those amino acids attach to the tRNA. Now remember, we said, again, if we go back here, right, the tRNA, right, here's your tRNA, has a specific amino acid that's attached here and a specific, anticodon that's going to match up with the codon here. So there's a specific process or specific enzymes that will attach the appropriate amino acids here based on what anticodon is there, okay? And essentially based on the codons they're going to match up with, right? Remember, let me go back. Let me just show you something real quick, and then we're going to come back, right? Remember, based on this genetic code. Okay, so if we talk about the genetic code, right? So U, right? In terms of that triplet codon, okay, that there is a specific amino acid that matches up with that phenylalanine, okay? And so what we're talking about here, again, is that we have to make sure the appropriate amino acid, sorry to go back again, the appropriate amino acid is attached here based on this anticodon, which is essentially is going to match up with that codon that's in that genetic code, okay? Hopefully I didn't just confuse you. We're going to come back to it anyway. Okay, so in terms of how that actually happens, right? So there are specific enzymes called aminoacyl tRNA synthetases, okay? Oh, by the way, just a reminder, you should have your review sheet with you right now, and you should be taking notes on it, whether it's electronically or handwritten. I prefer that you print it out and write it by hand because you can write drawings and do whatever you need to do on there, but I will be asking you to submit that again, okay? So you should have your review sheet. If you don't, pause it, get your review sheet, and start taking notes on it. Okay, so those specific enzymes are called aminoacyl tRNA synthetases. And there are 20 of them. 20 of them for each amino acid, right? So each of those 20 enzymes recognizes a single amino acid as well as that correct tRNA, okay? And so the process occurs in 2 steps. That amino acid gets joined to AMP, and it will form this aminoacyl AMP, and then that amino acid gets transferred to that 3' end of the tRNA, okay? So pretty simple. And so you can see this here, right? You have that amino, you have the amino acid, right? Joined to AMP up here. Okay, so the amino acid joined to AMP here, aminoacyl AMP, and then here's your tRNA, and basically it's going to attach that amino acid. Again, this is specific, right? So there is a specific enzyme for each amino acid, okay? And that enzyme specifically recognizes the appropriate tRNA to attach to, right? Because it has to make sure it's got the appropriate tRNA on this, the appropriate amino acid based on whatever anticodon is down here, okay? So again, it's sort of mediated through this AMP here. Again, the specific enzyme, right, is aminoacyl tRNA synthetases. So here I told you that both steps are catalyzed by that specific enzyme. Okay, so it's sort of a 2-step process. Again, we're sort of oversimplifying it, but showing you again the basics in terms of what's happening. So aminoacyl tRNA synthetases are the specific enzymes that add amino acids to the appropriate tRNA. Okay, that process or that reaction is actually 2 steps. Okay, the amino acid, first AMP is joined to the amino acid, then that amino acid gets transferred to the appropriate tRNA and AMP is released. Okay, so you should know the name of these enzymes and how this is happening. Oh, and again, something to draw your attention to. Remember, this figure 9.2, the 9.2 is corresponding to the last version of the textbook, okay? However, whenever I'm referring to the figures, I'm really referring to them from the PowerPoint, okay? But if you have the new version of the textbook and you're gonna be looking for this figure in the textbook, just look in chapter 10 it's the same figure. In fact, it's actually figure 10.1 in the new textbook. But bottom line is it's just almost exactly the same figure, okay? Okay, so it says amino acid is then aligned on the mRNA template by complementary base pairing, okay? All right, again, most of this stuff in here you probably know. Remember, amino acids are specified by more than one codon, most amino acids, right? Remember that that genetic code is redundant, right? Cells have about 40 different tRNAs for the 20 different amino acids, right? Because again, there are multiple codons that will code for the same amino acid. And so I'll remind you of that again when we look at that genetic code one more time. So some tRNAs, this is what I just said, right, can recognize more than one mRNA codon. Therefore, there are codons that will code for more than one, or there are multiple codons for one amino acid, okay? So we call that wobble, right? It's at that third codon position. So sometimes on that tRNA, right, G can actually pair with U, but it also can pair with a modified base called inosine. Okay, and also, sorry, G can actually pair with U, again, at this sort of wobble position to basically add this flexibility and this redundancy into the genetic code. Inosine, which is a modified base, I don't know if you remember, we talked about tRNAs, we said that they actually get processed before they're fully functioning. And one of the processing steps that occurs in a tRNA is that some of the bases are modified. And the reason why they're modified is because it allows for this wobble to happen. So this inosine, if it's in the third position, can actually pair with U, C, or A. Okay, and so that's how one tRNA, these tRNAs are able to recognize multiple codons. Okay, all right, so that's what this is saying here. If you look at that, this is what this is showing you. So this is saying here's phenylalanine, right? This is that amino acid. And if you look at the genetic code, right, for phenylalanine, you'll see that there are multiple codons that will code for phenylalanine, right? U, but also UC, okay? So again, phenylalanine, showing you two different triplets codons that can code for that same amino acid. And you can see that here, look at leucine, right? There's multiple codons that can code for leucine, okay? And for most of the amino acids there. So how does that actually happen? It's based on this. And so this is just showing you for phenylalanine, right? Phenylalanine, here's the tRNA, right? Here's the anticodon in the tRNA, AAG. And it's showing you this G pairing with C is what you would normally think of, right? Here, though, it's also showing you that, and so the codon then would be UUC here. And again, if you go look at this, right, UUC is one of the phenylalanine codons, right? But what else is UUU? So if we go back to that picture again, and we look here, we can see here's phenylalanine, right? Same anticodon here, but this G actually can pair with U as well, okay? And so that's where the sort of flexibility comes in. So again, two different codons here, right? Coding for the same amino acid. So when you look at the genetic code, this table, remember that these are codons in the mRNA, and right, codons in the mRNA, and what amino acid they code for. It's not the anticodon, these are the codons. So you can see them here. Again, it's the codon in the mRNA, sorry, over here. Okay, but you can see that this same anticodon can actually match up with both. Now, if you look at alanine here, right, which we're talking about here, right? So this is showing you some variability here. So U, C, U, right, is one of the codons. And so this is showing you kind of how this happens. So here's alanine, here's your tRNA. This one has GCI, inosine, in its anticodon space. Here's your codons, GCU, GCC, GCA. These are all options. And why? Because this inosine can actually pair with U, it can pair with C, and it can pair with A, okay? And so that's how, you know, you're sort of able to have this redundancy in the genetic code, okay? So when we say wobble, it's always in this third position. And a lot of the times the reason why this happens is because on the tRNAs, in this third position, you have a modified base that actually can bind with more partners than say that normal base, okay? So again, the modified bases in the tRNA allow for wobble to happen. So you can see here, this is the same tRNA, but it can match up with 3 different codons. Okay. All right. So again, I've already been referring to this a couple times. Remember that again, you have multiple codons coding for the same amino acid. Okay. Yeah, so that was the tRNA, right? So if we're talking about translation, we got to sort of set up all the pieces, okay? So we talked about mRNA, I feel like probably enough, mostly in the last chapter, right? The production of mRNA happens in transcription. We talked about how the mRNA gets processed. Remember, it gets a 5' cap, a 3' poly A tail. Introns are spliced out, right? And then it gets exported into the nucleus, out of the nucleus, and it's going to go find a ribosome. Okay, so mRNA obviously is an integral component of translation. So is tRNA, what we just talked about. Okay, we talked about the anticodon, the codons, the genetic code. Now we need to talk about the ribosomes. Okay, and so we name them according to their sedimentation rates. So sometimes remember, and we probably saw this before when we actually were talking about transcription of ribosomal rRNA, right? The different types. And so 70S, right? The S means sedimentation rate. So meaning when you put them in an ultracentrifuge, how it's sedimenting down, okay? So don't try to read into that any more than that, okay? So we call it 70S for bacterial and 80S for eukaryotes, okay? There's not just one ribosome, many ribosomes in a cell. So E. coli, about 20,000. If you talk about growing mammalian cells, they can have up to 10 million ribosomes, okay? And all ribosomes will have 2 subunits, a large and a small. And each of those subunits is composed of ribosomal RNA, right, rRNA, and also characteristic proteins that make up that subunit. Okay, so as you would expect, the subunits of eukaryotic ribosomes are larger, and they have more proteins than the prokaryotic ribosomes. In terms of the general makeup, they're similar, okay? So this is showing you the 70S ribosome for prokaryotes and the 80S for eukaryotes, okay? And these are the 2 subunits, right? Large and small. So in prokaryotes, you have a 50S and a 30S. And again, remember, these doesn't necessarily add up. The S has to do with sedimentation rate in an ultracentrifuge, okay? That's how we're naming them. Not like, it's not kilodaltons, it's not weight, because it's made up of ribosomal RNA and proteins, okay? All right. So within that 50S, that large subunit of the prokaryotic ribosome, you have 23S and 5S ribosomal RNAs. And then you also have 34 proteins, okay? So 2 different types of RNAs that make up this 50S subunit and also an additional 34 proteins. In the small subunit, you have the 16S rRNA and 21 additional proteins. For the eukaryotes, if you talk about the large and the small, you got a 60S large subunit and a 40S small subunit. The 60S is made up of a 28S and a 5.8S, and also the 5S ribosomal rRNA and about 46 additional proteins. Okay, here the 40S is an 18S ribosomal RNA plus 33 proteins. Okay, so you can see the makeup and essentially what each subunit is made up of, both ribosomal rRNA and different types of it, and a whole slew of proteins. Okay. All right, so this is just showing you ribosome structure, the large and the small subunit as well. And there have been many experiments that actually have shown that you can actually form a sort of in vitro ribosome, right? So it says ribosomes can be formed by in vitro, or can be formed in vitro. What does in vitro mean? It means in a tube, right? Talk about in vivo, it's actually in a live animal. In vitro, in a dish, in a tube. And so they can be formed by self-assembly. So basically, if you just put in purified ribosomal proteins in a tube and also ribosomal rRNA, they will sort of self-assemble into the ribosome, okay? And so you can really do these reactions within the tube and assess the function of each one of those proteins in those ribosomal RNAs, and that's what it's telling you here, okay? These ribosomal RNAs form characteristic secondary structures based on complementary base pairings, just like we saw with the tRNAs, right? They form that characteristic clover leaf or lowercase t shape based on complementary base pairing, okay? And then there is further folding that will result in other even more complex 3D structures. Okay, so these ribosomal RNAs, have a characteristic sort of folding and structure, and then they're associated with other proteins as well. Okay, so that's what this is showing you here. So if you look at this 16S, obviously it's a lot more complex than the tRNA that we looked at. So this is just the 16S ribosomal rRNA. And so let's go back and remind you where that is. That's that small subunit of the prokaryotic ribosome. Okay, so if you just look at that rRNA, not the other proteins that are there, just the rRNA, this is the structure of it. And again, all of these sort of secondary structures and shapes that are forming here, right, are due to complementary base pairing. Okay, so it says that it was first thought that ribosomal RNA only played a structural role in the ribosomes, right? And that basically the proteins that were there were the ones that acted as enzymes, right? And thus had catalytic activity. However, there were studies later that showed that the ribosomal RNA actually can act as an enzyme, right? It has catalytic activity. That's what this means. So in 1992, right, Knoller and his group showed that the large ribosomal subunit can actually catalyze the formation of peptide bonds even after 90% of the ribosomal proteins have been removed. Again, showing that really much of the enzymatic or catalytic activity in the large subunit is from the ribosomal RNA, right, and not from proteins. In 2000, there was also other evidence that showed if you look at, structural analysis of that 50S ribosomal subunit, you basically saw that it wasn't protein, it was ribosomal RNA. Okay, also another sort of line of evidence, right, that supports the fact that it's the ribosomal rRNA that really has much of the catalytic or enzymatic function in the ribosome. Ribosomal proteins are actually absent from the site of where the peptidyltransferase reaction occurs, right? So again, in terms of where those peptide bonds are formed, ribosomal RNA is responsible for catalyzing the peptide bond formation. It's not proteins that are there, okay? So there's been a sort of long, and this isn't every bit of evidence. There's lots of other evidence out there as well, but these are sort of three key points in terms of evidence supporting the fact that ribosomal RNA is really responsible for the catalytic activity. That's where the enzymatic activity is coming from, not from the proteins that are there. Okay, so this is actually showing you, if you look at the 50S ribosomal subunit, and again, let's go back and remind you what we're talking about, right? So if we're talking about the prokaryotic ribosome, we're talking about the large subunit here, right? The 50S. And if you look at what makes, up that 50S ribosomal subunit, the large subunit, you can see aqua is rRNA, purple is proteins. And so when you look here, right, this is essentially the sites, right, the E, the P, the A site, where essentially translation is happening, right, and the peptide bonds are being formed, and basically that growing polypeptide chain, the growth of the polypeptide chain is occurring. You can see that it's mostly rRNA there, not protein. Okay. All right. So again, that's just sort of following up. Ribosomal proteins play largely a structural role, and that really the rRNA functions as a ribozyme. Okay. And so, and we've talked about this a little bit before in terms of, you think about evolution, right, and evolutionary implications. RNAs are thought to have been the first self-replicating macromolecules, right? Those RNAs could act as genetic material, but also as enzymes, right? And so they could catalyze their own replication because they had that catalytic or enzymatic activity. Okay, so it says the role of and also the role of rRNA in the formation of peptide bonds provides more evidence that they had catalytic activities beyond just replicating themselves, right? So they could have acted as enzymes for other reactions in the cell as well. And so this provides more evidence that really maybe RNA was around sort of first as that first molecule or biomolecule, okay? All right. So it says here, most eukaryotic mRNAs are monocistronic, meaning that they encode a single protein. Okay. A lot of prokaryotic mRNAs are polycistronic, and that means they encode multiple proteins. Okay. And so each of which is translated from an independent start site, but the mRNA itself can code for different mRNAs, right? So proteins. So one mRNA, multiple proteins in prokaryotes. In eukaryotes, usually it's monocistronic. However, we did talk about like alternative splicing and things like that, which leads to the production of other proteins, but that's sort of a different kind of process. So don't kind of, I guess, confuse that. But obviously you can have mRNAs where you do have alternative splicing that then will code for a protein. But essentially the alternative splicing kind of makes different mRNA molecules, right? That then code for those different proteins. So that's the idea here. Okay, so for prokaryotes, you have that one mRNA, and actually you can encode for multiple proteins just from that one mRNA molecule. All right, so just again, and if you need to kind of go back and review the sort of alternative splicing to sort of make you understand that, go ahead and go back. But this holds true even in the light of alternative splicing. Okay, so eukaryote mRNAs mostly are monocistronic, encode a single protein. Prokaryotic, polycistronic, encode multiple proteins. Okay, it says mRNAs have non-coding untranslated regions that are called UTRs at the ends. Okay, and so here, if we look, this is basically illustrating what I just told you in the last slide. Right, in a prokaryotic mRNA, here's one mRNA molecule. You know, depending on when translation starts here, it makes protein 1. Translation that's going to start here makes protein 2. Right here, protein 3. This is one single mRNA molecule. Eukaryotic mRNA, it's really one single translation start site. There are not multiple translation start sites. Okay, one protein, and it's just showing you these UTR regions as well. Okay. In terms of the process of translation, right? So in both prokaryotes and eukaryotes, translation always starts with methionine, okay, which is usually encoded by AUG in terms of the codon. And so that, it says the signals that identify initiation codons are different, though, in prokaryotes and eukaryotes, okay? So the codon that starts it, that's the initiation of translation, or the start site, is AUG, and codes for methionine, that's the start. However, what recognizes them is different, okay? So initiation codons in bacterial mRNAs are preceded by something called a Shine-Dalgarno sequence. And again, you've probably heard of these before in micro. And that will align the mRNA and the ribosome. They can initiate translation at the 5' end of an mRNA, and also at internal initiation sites, okay? Because remember we just said that they're polycistronic, right? There can be internal translation start sites. Okay, for eukaryotic mRNAs though, they're recognized by the 7-methylguanosine cap at that 5' end. Remember we said mRNAs have to be processed in a certain way, okay? One of the ways is the addition of that 5' cap, and it's important in the initiation of translation. Okay, so once that ribosome kind of recognizes that 5' cap, the ribosomes can then scan downstream and they'll find the initiation codon. Okay. Okay, so for prokaryotes, for initiation of transcription, you have to have that Shine-Dalgarno sequence. Okay, so that'll align the mRNA and the ribosome. For eukaryotes, it's really that 5' cap that sort of gets the ribosome there, and then it will scan through for that initiation codon. Okay, so that's what this is showing you here, right? Here's your prokaryotic mRNA, and again, this is the 16S rRNA, essentially part of the ribosome that's going to recognize this Shine-Dalgarno sequence, right? Recognizes it, and then that's what's going to essentially, we're going to then start translation sort of downstream from here. For the eukaryotes, okay, here's your, again, the small subunit, eukaryotic ribosome, it recognizes that 5' cap, okay? And then we'll scan down till it finds that start site, okay? So again, you can kind of see here, here's the Shine-Dalgarno sequence, and then downstream a little bit more will be that start site. Okay, So if we talk about the basic stages of translation, we've probably talked about the basic stages of translation before. Again, we get in a little more detail here. So initiation, just kind of started talking about that, right? Elongation, although we're going to talk about it more. So this is kind of what we just talked about here is sort of what's going to signal for translation initiation. Now we got to talk a little bit more detail about that initiation, okay? So initiation, elongation, and termination. So it says a specific initiator, methionine tRNA, right? So a tRNA that codes for methionine that recognizes that AUG will, well, along with the mRNA, will bind to that small ribosomal subunit. Okay, so the small ribosomal subunit comes in contact again with the mRNA and the initiator tRNA first. Okay, then once those three things all associate together, the large ribosomal subunit will then join, and basically that will form that functional ribosome. If the large ribosomal subunit never joins, there's no translation that's going to occur. Okay, so you can think of the small ribosomal subunit, you know, as being the one that kind of is the sort of, I guess, initiator, right? Is there to sort of get the mRNA in the right place and the tRNA all together, and then the large ribosomal subunit comes in second. Obviously, then also a whole bunch of proteins will come in as well, okay? All right, so again, this is just showing you basics here. So here's your initiation. Remember the small ribosomal subunit joins first, associates with the mRNA, and also that initiator tRNA, which isn't really showing you this here, right? And then you got the large subunit, and then elongation will occur, meaning elongation of that growing polypeptide chain, right? And then eventually termination, everything will basically dissociate, okay, because you're going to reach that stop codon, and the polypeptide chain will be released. Okay, so that's super general overview. We got to talk a little more detail. Okay, now, so again, like I said, we got to talk about initiation. Okay, we haven't even really talked about the details of initiation. So if you look here at this table, it's showing you some translation factors. Okay, so in terms of the role, all right, so first you look at initiation there. And you can see some of the factors involved in initiation. Yeah, it looks like they kind of got rid of, they didn't have this table in the new version of the textbook. I was just kind of chucking there. And nobody needs to memorize any of this, but sometimes it's nice just to kind of see that there are many. And you know what? There are probably many more right now, okay? Anyway, initiation, not maybe many more, but there may be a few more that have been recognized. But like I said, nobody needs to memorize anything. In fact, if you look at the names, right, it says IF, initiation factor 1, 2, 3, prokaryotes, right? If you talk about eukaryotes, it's little e for eukaryote, IF, and then numbered and lettered from there, okay? So know that if you see IF, it's an initiation factor, right? If you see little EIF, it means it's a eukaryotic initiation factor. If you likewise see EF, it means it's an elongation factor, okay? Meaning it's a protein that's going to be involved in elongation. Here, same thing, little e, big EF, eukaryote, okay? Termination, RF, because release factor. All right, so just to kind of just be familiar with the terminology, please don't memorize any of these. Okay, so in terms of initiation, if we talk about initiation bacteria, okay, as I said, that, you know, that small ribosomal subunit is going to come into play first, right? So the 30S ribosomal subunit bound to these two initiation factors first, right? IF1, IF3, then mRNA, that initiator tRNA, okay, and IF2 will bind, okay, and then these two initiation factors will be released, and that large subunit will come in, okay? So you can see this here. So the idea here is to just understand that there are many proteins involved in this, okay? And so this is just showing you initiation and showing you some of the proteins involved, right? So here you can see, again, what I just mentioned on the last slide. Here's your 30S subunit, these two initiation factors associated with it. Here's your tRNA, right? Your methionine tRNA. That's going to match up with the the AUG in the codon, right? That start codon. And here's another initiation factor that's going to come in. These all associate with the mRNA. and then that large subunit comes in, okay? And essentially then it's going to be able to start. Okay, so in eukaryotes, that initiation is more complex. Okay, so it's the same idea though. It's just that there's more initiation factors. Okay, so this is showing you in here. Again, nobody needs to memorize this. It's just to sort of appreciate the sort of complexity of this. Okay, so it's the same idea. Your small subunit is associated with particular initiation factors, which are proteins, right? Here's your initiator tRNA. Okay, they're all going to associate together with the mRNA. And you can see here, remember that I told you that in eukaryotic translation or initiation, you know, this complex or the small subunit, along with all these proteins, right, really has to recognize that 5' cap, okay? And so that's what this is showing you here, and that you sort of have this, some of these sort of proteins. This is kind of showing you this sort of looping here, where you can see kind of how these proteins can sort of associate with the cap and also some of the other mRNA there, okay? So yeah, so again, you see here, this is that 3' poly A tail. So there's a specific protein that binds to this poly A tail, this PABP, that then associates with the other initiator proteins here, okay? Again, you guys don't need to know all these details, but I'm just sort of pointing out a few things that are kind of important in the process or sort of unique. Remember, we talked about the necessity of that 3' tail. We said that it sort of protects against degradation, but it actually has a specific function in terms of binding a specific protein involved in the initiation of translation. Okay, so again, if that 3' tail is not there, that mRNA is not going to be able to be translated. All right, and so again, you can see now here's your small subunit and a whole slew of proteins along with that initiator tRNA that are all bound there, that mRNA. All right. And so it's showing you here, it sort of scans until it's going to find that AUG, right? Once it finds that AUG, that start site, all of these proteins are released, okay? And then the large subunit comes in and translation can carry on. Okay, so again, that was all initiation, okay? It's similar between prokaryotes and eukaryotes, but slightly different, right? In eukaryotes, that 5' cap isn't involved, right? I mean, sorry, in prokaryotes, no 5' cap, right? So this is initiation in bacteria. Because remember, it can start translation internally. So it's not tied to that 5' cap because that wouldn't make sense. If it was, it wouldn't be able to have these separate initiation sites internally, okay? So for some differences between the two, bacteria or prokaryotes, the 5' cap isn't involved in initiation. However, the sort of process is very similar, right? That small subunit associated with proteins, right? And the initiator tRNA, they all bind to the mRNA, find that start site. Once that start site's found, those initiator or initiation factors here, or initiator proteins, however you want to think about it, they go away and allow the large subunit to come in. Okay, similar process in eukaryotes, except that there are more proteins involved, and also this 5' cap is involved, and that's where this all sort of associates together at first. Then the whole thing moves down to scan, right, for that start site. Okay. All right, so that's initiation. Initiation involves, if I asked you sort of to list some of the things that are involved in initiation, right? Initiation factors, the small ribosomal subunit, the initiator tRNA, right? The start site or the start codon, right, is AUG, okay? So, I mean, those are some things that we should be able to remember. All right, so now if you're going to talk about elongation, now we're talking about that elongation of that polypeptide chain. So now you got everything set up. The mRNA is there, right? The small ribosomal subunit is there. The large one is there. That whole complex is all, associated together and basically made up of all the components, right? All the components are there. Okay, so it says it's very similar in both prokaryotes and eukaryotes. So that large ribosomal subunit, or the ribosome in general, has 3 binding sites. P for peptidyl, A for amino acyl, and E for exit, okay? The initiator methionine tRNA will bind to that key site first, okay? Then the next amino acyl tRNA, meaning that next amino acid that's going to come in, right? The tRNA with the next amino acid will bind to the A site by pairing with that second codon in the mRNA. So it's getting ready, right? It's that next codon in line. There are also other proteins that are going to help out with this, right? So remember I showed you that table that there are elongation factors. So the initiation factors, the elongation factors, they're proteins, okay? You don't need to memorize any of the names. All right. And so this is what this is showing you here. Again, you first have that start codon here, your initiator tRNA, which codes for methionine, right, in the P site, bound the anticodon, essentially matching up with that codon there. The next one in line, right, the next tRNA that's carrying that next amino acid, based on whatever codon is here, is going to come in at that A site. Okay, so it comes in at the A site. And again, you can see how there are other proteins, particularly showing you here, these elongation factors involved in bringing that tRNA to the appropriate site, to the ribosome. Okay, so you can kind of think of this elongation factor as being a helper, sort of a transporter to get that tRNA where it needs to go. All right, so we move on. And so now we can see here in this is the A site, right, the P site. Okay, we have the initiator. Now we have the next one in line, okay? And basically you have the peptide bond that's going to form, okay? So right here, peptide bond forms between those two amino acids. Okay, got that here. And then basically you have translocation, right? So you can see here that this guy, right, this guy here now has both the alanine and the methionine bound to it, right? And it is going to translocate into the P site and then move this guy, the original initiator tRNA, into the E site, E for exit, and that will leave. Now there's an empty space for the next tRNA with the next amino acid to come in. And you can see here that there are other proteins, other elongation factors that are involved in the process of translocation, okay? I'm not going to ask you specifically what types of elongation factor, what specific elongation factor is involved in translocation, but understand that there are proteins here that are helping with that process, okay? We should, again, know that if it's something that says, if it says EF, it means it's elongation factor. It is a protein involved in the process of elongation, okay? If we see IF, it is a protein involved in the process of initiation of translation. Okay, so this is a lot of words on the slide, so I'll apologize in advance here. So it says the selection of the correct amino acyl tRNA determines the accuracy of protein synthesis, obviously, right? If the wrong amino acyl tRNA is there, it would put the wrong amino acid, okay? So it says base pairing alone actually doesn't account for that. So there is something in the small ribosomal subunit that actually helps with this and helps increase the accuracy. And so you can call it a decoding center. So it says it'll recognize the correct codon and anticodon base pairs and basically will discriminate against mismatches, okay? So there is a conformational change that will happen if the correct amino acyl tRNA is there, okay? And so if it's not the correct one, that conformational change won't happen, and therefore it would sort of be booted out, okay? And so just remember that there is sort of more, it's more than just the matching of the codon to the anticodon. There is a sort of part of that small ribosomal subunit sort of has this extra check to make sure that that is the right one. Okay. All right. All right, so we said all this. I showed you all this from the picture here. Okay, so translocation, right? We said all this. So basically, there's a lot of words on these slides here, but it's describing what we just went through in this picture here. Okay, we talked about elongation. So the words surrounding those slides essentially are describing what I already went through with you. Okay. All right, I think everything. Yeah, I already said all this stuff. All right. Okay. All right, so there's one kind of little side note here, I guess that's worth talking about. It says as elongation continues, okay, this elongation factor 1 alpha, or you can call it or EF2 if you're talking about prokaryotes, is released from the ribosome bound to GDP. And in order for it to function in elongation again, it actually has to be reconverted into its GTP form. Okay, so there's other proteins or other elongation factors, and you saw them in that table that I showed you, that actually are involved in just converting this elongation factor that's bound to GDP back to its GTP form. Okay. And the reason why, again, it's sort of worth mentioning is because this is sort of a level of regulation of proteins, right? So it says regulation of this elongation factor 1 alpha by GTP binding, right? And hydrolysis, meaning GTP being converted into GDP, is a common method of protein regulation. Okay, so proteins bound to either GTP or GDP is one way in which the cell can regulate the activity of the protein. Okay, meaning that if the protein is, say, bound to GDP, it may be in its inactive form, right? And see this here. So here's so this is the reason why we're going through this, right? So meaning, again, if this elongation factor is bound to GDP, that's its inactive form. If there is no mechanism to convert it back to its GTP form, it will not be active. Therefore, it won't be able to be involved in elongation, and translation won't be able to occur, right? And this is not only true for these types of proteins or this specific protein here, it happens a lot in the cell, right? So again, you can think of any given protein as potentially being able to be bound by GDP or GTP. And if it's bound by GDP, it's inactive. It only would be active when it's bound to GTP. Okay, so these steps are super important in sort of the regulation of the protein. Okay, and super important specifically to this process here. Again, if this EF1 alpha is not converted back into the GTP form, okay, it's not going to be able to participate or catalyze that sort of elongation process in translation. Okay, so this elongation, right, will continue until a stop codon is reached. Okay, so UAA, UAG, UGA, those are all stop codons. Okay, and so once that stop codon gets translated into the A site, and once that stop codon is in the A site, then you don't have a tRNA that comes in, a release factor will actually recognize those signals. Okay, so again, we saw that from the table. We saw RF is a release factor. So if I ask you, what is RF involved in? Initiation, elongation, or termination, we should know that it's termination, right? Okay, all right. So it's saying here, prokaryotes, right, RF1, in eukaryotes, it's little e RF1, okay? So here in prokaryotes, there's actually, right, in terms of what recognizes what, RF1 will recognize these two stop codons, RF2 recognizes this. In eukaryotes, the same one recognizes all three stop codons. Okay, so we see a picture here. Once this stop codon gets into the A site, right, what's going to bind here? Not another tRNA, but a release factor. So that that release factor recognizes the stop codon, binds, and essentially it's going to cause release of that polypeptide chain, and then eventually it will cause all of this complex to disassemble. Okay, so everything disassembles, and then essentially, obviously, these ribosomal subunits will then go and translate another mRNA. Okay. The other thing is that if you think about one mRNA, it can be translated simultaneously by several ribosomes, meaning one's working its way down while another one can then bind and basically make multiple copies of that protein. Okay. And so we call that a polyribosome or a polysome, when you basically see one mRNA molecule with a whole bunch of ribosomes bound to it, right? So these are a whole bunch of ribosomes translating this one mRNA molecule that's sort of under here that you can't really see. All right, so again, sort of super basic here, and this is just showing you a drawing of that polysome. Here's one mRNA molecule, and here are multiple, right? So this one has pretty much already translated the whole thing, right? This one kind of just started. And so basically, once all these are done, you would have 4 copies of the same protein, right? Okay, so in terms of regulation, actually, you know what? I think I am going to, let's see, I may stop here and then just start again. Yeah, that's probably a good idea. We're just about halfway through, okay? So that's what I'm gonna do. I'm gonna stop here, and then there will be a part 2 to this lecture, to which we're gonna then talk about regulation of translation and protein processing, okay? Oh, sorry, I should have ended the recording.